Jonathan White, Bio 260, online lab exercise for biochem part 2. This is the TSI slant number 13. I need to obtain 1 TSI slant right over here. Make sure not to use a dehydrated slant. If the slant reaches close to the bottom of the tube, this is how you know it is dehydrated. I need to label my tubes and then inoculate the needle in the micro incinerator for about 10 seconds. So I'm going to take that needle, place it in here for about 10 seconds, take the source culture here, remove the cap of it, place the source culture near the micro incinerator, sterilizing the lip of this for 2 to 3 seconds. And then with my sterilized needle, take a small sample of the source culture. So placing it gently and pulling it back across the same line. So now the needle has a small sample on it. Then you're going to take the source culture and you are going to sterilize the lip of it again for 2 to 3 seconds. Recap it and place it back. Then I'm going to need to take one of my pre-labeled TSI slants right here. I am going to uncap it. I'm going to sterilize the lip of it for 2 to 3 seconds. And then stab the tube deep into the butt of the tube, or stab the needle deep into the butt of the tube. So you're going to come down the line here, and then withdraw the needle from the butt, streaking a single line up the slant. So then along the same line, streaking a single line up the slant. Now that I've done that, I'm going to take the tube and I'm going to hold it next to the micro incinerator, the lip of it, and sterilize the top of it for 2 to 3 seconds. Recap it, place it back. Take my needle, sterilize it in the micro incinerator for about 10 seconds and let it cool off. And then with the TSI tube, the triple sugar ion test, you are going to need to incubate it at 37 degrees Celsius for about 24 to 48 hours. You must read these results by 48 hours. If you're unable to do so, you have to incubate your tubes at room temperature to slow the growth.